We are living in an era where human DNA is being programmed, debugged, and updated exactly like computer software. In hospitals right now, artificial intelligence is designing genetic edits in real time, and the FDA has already approved therapies like Cas9, which permanently rewrites the genetic code of patients suffering from sickle cell disease. Recently, a newborn known as baby KJ became the first infant saved by a rapid, personalized CRISPR therapy. Doctors sequenced his specific mutation, designed a custom edit, and delivered a cure in a matter of weeks. These milestones prove that gene editing has crossed the threshold from a slow laboratory experiment into an active, scalable technology rewriting human health. To understand how we got here, we have to look back to the 1990s. Researcher Francisco Mojica was studying bacteria when he noticed a strange pattern in their DNA, a rudimentary adaptive immune system storing snippets of invading viruses to fight off future infections. By 2012, scientists Jennifer Doudna, Emmanuelle Charpentier, and Feng Zhang figured out how to hijack this bacterial defense system and reprogram it for human use. CRISPR relies on two components. First, a guide RNA acts like a molecular search bar, scanning the genome until locking on to a specific target. Once found, Cas9 arrives, acting as molecular scissors, clamping down to create a blunt double-strand break, severing the DNA helix. After the cut is made, the cell panics and tries to repair the damage. It relies on internal patching processes, gluing the broken ends back together in a messy way, or using a provided template for a precise fix. While revolutionary, relying on those blunt double-strand breaks can cause unpredictable off-target errors. If we want to safely edit humans at scale, we need a hardware upgrade. That upgrade is CRISPR 2.0. Scientists have developed a new generation of precision tools, base editors and prime editors, designed to fix the bugs of version 1.0 by avoiding blunt double-strand breaks entirely. Panel B illustrates base editing. A modified Cas9 and enzyme act like a chemical pencil, swapping a single DNA letter without cutting. Prime editing, panel C, acts as search and replace. A Cas9 nickase and reverse transcriptase write new sequences directly into the target. By replacing destructive cuts with exact, predictable writing, these tools have made gene editing significantly safer and far more reliable. But the best genetic software in the world is useless without a reliable physical drive to install it into human cells. Historically, researchers relied on modified viral vectors to deliver gene therapies, which were difficult and expensive to manufacture. Today, the standard has shifted to lipid nanoparticles, or LNPs. Using the exact same delivery technology proven in mRNA vaccines, these microscopic fat bubbles can safely parachute CRISPR components through the bloodstream, fusing with cell membranes to release their payload directly into target organs like the liver. On the software side, the 2026 upgrade includes deep integration with artificial intelligence. AI copilots like CRISPR GPT have moved from theoretical concepts to functional laboratory tools. They instantly automate guide RNA design, predict potential off-target mutations, and plan experiments under human oversight. The combination of LNP delivery and AI-driven design has drastically shortened the time it takes to build and test new therapies. Gene editing has moved from a bespoke artisan craft into an industrial manufacturing phase. This industrial speed is what makes cases like baby KJ possible. At the Children's Hospital of Philadelphia, his medical team relied on these rapid design cycles to manufacture a custom therapy. The case demonstrated that an individualized N-of-1 CRISPR therapy can be sequenced, manufactured, and infused into a patient within a single month. Building on years of advocacy for ultra-rare disease frameworks, the FDA used the data generated from KJ's treatment to formalize a new regulatory pathway, the plausible mechanism pathway. Under this new framework, bespoke gene therapies targeting understood mutations can bypass traditional, decades-long clinical trials. Instead, they rely on proof of mechanism and real-world evidence. For the first time, regulatory laws are updating to match the iteration speed of software, permanently clearing the bottleneck, keeping cures away from patients. Broadening the scope, this ability to rewrite biological code is now reaching the planetary level, entering global ecosystems and agriculture. In agriculture, scientists are using a technique called gene stacking to rapidly engineer climate-resilient crops. We now have varieties of heat-tolerant wheat and modified barley that reduce methane emissions when fed to livestock. Researchers are also deploying gene drives, a mechanism that forces a specific genetic trait to spread through wild populations. This gives us the ability to suppress or entirely eradicate devastating pests, like the mosquitoes responsible for malaria. CRISPR has transformed biology into an information technology. We possess the tools to read, debug, and rewrite the source code of life, securing human health and engineering our planetary survival.